Okay, so for the continuation of our non-fermentative gram-negative bacilli, we will be discussing Moraxella, Oligella, and Cyclobacter. Moraxella species, okay, so these are considered to be as the normal flora of our mucous membrane. So meaning to say, our mucous membrane are actually tinged with this kind of bacteria, and they are strictly aerobic. So the requirement, there's an immense requirement for oxygen. So you need a very high oxygen concentration. And if you will be looking at the morphology, so they are bacilli up to the bacilli level. No? Excuse me. They are also strongly catalase positive, oxidase positive. They are non-motile and generally biochemically inert. So when we say inert, it means that they are inactive with regards to carbohydrate oxidation. So the term asaccharolytic, I think I have already uh, explained this to you. So the term asaccharolytic refers to any organism or any organisms that cannot ferment any sugar at all. So if the organisms cannot ferment any sugar at all, they cannot break down complex carbohydrates, then they are actually termed as asaccharolytic. So this is an example of Moraxella. So you would notice that they are actually a cocobacilli. Okay, so there are actually two popular species, and one of them is Moraxella catararis. So the most frequently isolated and species, and as what I told you, they are actually they are actually uh, normal flora of the mucous membrane. So that's the reason why they are very much common in respiratory and ear specimen. Okay, so the problem with Moraxella, not really the problem, but what makes it more confusing for Moraxella is that they are often mistaken for Neisseria because some of them would really seem to appear like cocci. So if isolated from the mucous membrane, and also they are also oxidase positive. So it's quite really difficult morphologically to distinguish them from Neisseria. But except take note that Neisseria can ferment glucose, but Moraxella cannot ferment any sugar at all. So I think that's one of the things that you really have to look into if you are in the laboratory and confused between Moraxella and Neisseria. And then Moraxella lacunata is a common conjunctival isolate. So which means that these are isolated from the eye or from the eye secretions. Okay? So we also have Moraxella non-liquefaciens and Moraxella osloensis. So the second most commonly isolated species of Moraxella is Moraxella liquefaciens. And these particular organisms do not, or does not typically grow on Conky agar. And they most of the time reside as a normal microbiota of our respiratory tract, though it's very rare that they can cause diseases in humans. Now, another species, perhaps the reason why I put them together is that Moraxella osloensis is morphologically and biochemically similar to Moraxella non-liquefaciens. Although Moraxella liquefaciens are mostly of the time found in respiratory tract, but Moraxella osloensis is actually found at the genitourinary tract. So though they are biochemically and morphologically the same, the site where you mostly isolate them would be actually different from one another. Okay, and then Oligella. Okay, so Oligella is a small paired. So as you can see here, this one is paired. Okay? Bacillary, gram-negative bacillary or coccoid organisms, and they are most often isolated from the urinary tract. So the organisms do not grow on McConkie agar. So the one that we have here is a chocolate agar plate, and this is a blood agar plate. So they do not usually grow on McConkie agar. They are non-oxidative. They are FAD positive, okay, or PDA positive, meaning to say they are able to remove amino acid from phenylalanine because that's the purpose of the phenylalanine deaminase. Deamination of phenylalanine means that removal of amino acid. Okay, there's oxidase positive, nitrate reduction positive, and they are capable of gas formation. So usually susceptible to most antimicrobials, though recently some resistance has been reported already. And then we have species of Oligella here, and these are Oligella ureolytica and Oligella urethralis. So the term Ureolytica obviously means that these particular organisms are able to break down urea, hence the term Ureolytica. So most strains are motile by means of Peritrichus flagella, and they are PDA positive. So reported to cause bacteremia in patients with AIDS, meaning to say there's a tendency for this particular organism to become opportunistic. And it has also been reported that an 18-month-old child with pneumonia has Oligella. So they were able to isolate Oligella ureolytica from an 18-month-old child with pneumonia. Now, obviously, the species Urethralis refers to the urethra, a part of the genitourinary tract, primarily because Oligella urethralis is a commensal of the genitourinary tract. Okay, now let's move on to another genus. This time, we are discussing Cyclobacter species. So it's not really a popular genus, but there are 34 species. Okay, 34 species have been identified already. And most of these were actually isolated from fish, processed meat, and even in poultry. So to identify, if you remember our introduction, so when you say psychotropes, this particular term of the one that we used during the introduction, this means the bacteria is cold loving, a psychotropes. So this is probably where Cyclobacter got its name because the organism can grow as low as 5 degrees centigrade up to 25 degrees centigrade. So if we're looking at the temperature range of Cyclobacter, this is between refrigerator temperature up to room temperature only. And they can rarely grow at 35 degrees centigrade. Hence the term Cyclobacter. So the organism is also nitrate positive and can grow on tire Martin medium. So the organism is also non-motile. They are oxidase positive, and we consider them as an oxidative diplococcus. Okay, so they are oxidative diplococcus. So when you say diplococcus, they are usually seen in pairs, such as in this case. So here's another example of in pairs. Okay. So it has also been reported that they can emit a rose odor, and these bacteria are usually susceptible except to penicillin. So the implication that a bacteria can grow as low as 5 degrees, meaning to say these organisms can probably be implicated in food spoilage. So this explains why food, even if they are stored at refrigerator, can still be spoiled. Not to mention that these organisms are isolated from fish, processed meat, and poultry. Okay, so that is Cyclobacter. So the most frequently isolated species of Cyclobacter is Cyclobacter sanguinis and Cyclobacter phenylpyruvicus. So these two species were actually recovered from the eye of a newborn who acquired the infection nosocomially via a water source. So can you just imagine that this newborn was in the hospital and because of the contaminated water, so this newborn got infected with Cyclobacter. So recovered also from the blood and the cerebrospinal fluid of a 2-day infant. So can you imagine they collected CSF from a 2-day infant? Wow. And these particular organisms were isolated from the urine and genitourinary tract. Okay, so that is Cyclobacter sanguinis and phenylpyruvicus. Okay, so let's now move on to the less commonly encountered non-fermentative gram-negative bacilli. Okay, so we have here Alkaligenes and Chromobacter. Okay, so these two genera are found in water and are resistant to disinfectants. Wow, can you imagine, no? They are resistant to disinfectant. So, having said that, it is not surprising to know that they are isolated in specimen from hospitalized patients since they are resistant to disinfectant. So, what kind of specimen? So, this would include urine, feces, sputum, and even probably wound swabs. Okay, so they are a saccharolytic member. Okay, and they're less likely to be isolated from clinical specimen. So they can grow well in most media, including McConkie agar. So this is how they look like in sheep's blood agar. So they are non-pigmented. So therefore, the term "a" refers to the absence of, chromo refers to color. Since these particular organisms are non-pigmented, okay, so a Chromobacter is actually a clue that you would probably get that you won't expect any pigment at all for these particular organisms. Though some strain may produce a fruity odor and a green discoloration. Okay, but that particular green discoloration is not necessarily a pigment. Okay, so these are example of colonies of Chromobacter. Okay. So microscopically, this is how they look like. So they're gram-negative bacilli. So Alkaligenes fecalis, that's one species, and a Chromobacter species possess Peritrichus flagella. So when you see Peritrichus flagella, the entire bacterial cell is actually being surrounded by flagella. So that's what we meant by Peritrichus flagella. So flagellar stains, therefore, would become very useful for identifying this kind of bacteria. Now, for the biochemical reactions and treatment, so they cannot oxidize any tube in OF medium with deep blue color at the top, except for one species, and that is the alkaligenous psilocyclidans, in which these particular organisms are able to produce acid, okay, by oxidizing glucose and xylose. So they are able to reduce nitrate to nitrite. And not special mention for alkaligenous fecalis because it will further reduce nitrite to nitrogen gas. Okay, so treatment varies by isolated species. So usually, you really have to perform antimicrobial susceptibility testing to help clinicians in choosing the appropriate antibiotics. And antibiotic resistance, just like any other bacteria, has also been increasing for this kind of species. So moving on, we also have another less commonly encountered non-fermentative gram-negative bacilli, and this would also include Brebon diminuta. Okay, so Brebon diminuta is an infrequent isolate. So it's not every day that microbiologists would be encountering this kind of bacteria, but they have been found in blood, CSF, urine and wounds, and considered to be as most of the time a contaminant. So recently considered to be a pathogen, and they actually carried the VIN2 metallo-beta lactamase. Wow. So what is the implication of having this particular gene? So if they carry VIN2 metallo-beta lactamase, they would definitely contribute to multi-drug resistance. Okay, so Rapundimonas can grow on Maconti agar. So this is a Maconti agar, and you would notice that they would produce a colorless or white colonies because they are a non-lactose fermenter. So they are slender, gram-negative rod. They are motile and possess a single polar flagellum. So they can oxidize glucose. They can oxidate, and they are oxidase positive. And unfortunately, they demonstrate resistance to numerous antimicrobial agents. So, though they are very rare, you do not want to get infected with this bacteria because of its resistance to many antibiotics. So, one of the most commonly encountered species is Brebundimonas, Brebundimonas vesicularis. Okay, and these particular species are reported to cause meningitis, infective endocarditis, and infections in CAPD patients. So when we say CAPD, this stands for chronic ambulatory peritoneal dialysis. So these patients are still able to walk, but they are most of the time in constant dialysis. So um, I've known person, I know a person who is in dialysis at least 3 times a week. So that person is considered to be as a CAPD patient, and they are actually at they are actually predisposed to have Prebundimonas fascicularis. Only 25% will grow on Maconti agar, but if ever that they are able to grow, most strains are able to produce orange intracellular pigment. So the one that you can see here, the orange intracellular pigment. And they are oxidase positive. So this is the filter paper. So you get the colony of bacteria, and then you add oxidase reagent, and if it turns black, purplish, and then black, then that's considered to be as an oxidase positive. So they can oxidize glucose and maltose, and they are susceptible to several several antimicrobial agents. Okay, so Rebundimonas species, phylasculine hydrolysis is actually very useful for this organism because 88% of Rebundimonas vesicularis isolates are positive. So, so this could most probably be Escularis, and this would probably be diminuta. Okay? Rebundimonas diminuta are rarely positive with the bile esculin hydrolysis test. Now, CDC has actually assigned EO, which stands for Eugonic oxidizer. Eugenic oxidizer. Okay, so Sorry for that. Okay. So EO stands for Eugenic oxidizer. Though there are several Eugenic oxidizer, the EO3, EO4, but before that, meron pang EO2. Okay, so however, EO2 has now a name. So it is now Paracoccus yi I. Yi I. Okay, so it's a bizarre name. So members have been associated with human infection, but their clinical significance remain unclear. So, so they are oxidase positive, non-motile, saccharolytic coccobacilli. Um, no, sometimes they do not grow on McConkie agar or ever there's a growth, it's a very weak. So they can oxidize glucose, xylose, and some have actually pigment. Okay, so that is the EO. So EO is a genic oxidizer. Okay. So as you can see in the morphology, so um, they are also a Coccobacilli, and these Coccobacilli are most of the time in pairs. So Diplococcobacilli. So that's how we describe this particular info. So this one is another pigmented bacteria, Chromobacterium violaceum. Okay? And this particular bacteria is isolated from wound contamination with soil and water. Okay? And as you can see, the reason why they are called violaceum is because of the violet pigment that this bacteria can produce. And the violet pigment is actually due to violacine pigment. Okay? And these organisms are opportunists, which means that they attack immunocompromised patients with neutrophil deficit, and they are seen in cases of osteomyelitis, in abscesses, septicemia, UTI, and gastrointestinal tract infection. So skin lesion is a typical port of entry, and the organism is fermentative. Okay, gram-negative bacilli, oxidase variable motile usually produce violet pigment, catalase positive, and indole negative. And do you know why they are able to produce violet pigment? And the reason why they're able to produce violet pigment is actually due to quorum sensing. And quorum sensing means that whenever bacteria whenever bacteria are in high number, quorum means increase in population. Okay, they become more virulent, and one of the things that they are producing is a pigment such as violacine pigment of Chromobacterium vellulaceum. And usually, in order for us to optimize the production of the violet pigment in Chromobacterium vellulaceum, we are using a Luria bertani medium. So Luria bertani medium would actually optimize. Okay, and because of this characteristic, the Chromobacterium vellulaceum is most of the time being used as the screening organisms or the test organisms. We are using them as test organisms to screen substances for quorum sensing activity. So for example, I have isolated a certain compound, and for example, a compound from a plant. So if I want to know if this compound can would have a anti-quorum sensing activity, I would be using Chromobacterium villasium as the test organisms. If the pigment disappears, and then this particular compound would indeed have anti-quorum sensing activity. But if the organisms died, they do not have, or these organisms do not, or this compound do not have anti-quorum sensing activity, but they probably have antimicrobial property, but not anti-quorum sensing activity. Okay? So that's the reason why it's a very good thesis or research to screen for compound and to check whether they have quorum sensing activities or not. Okay, although it's quite difficult now to acquire Chromobacterium violaceo, because if you want to do a thesis regarding screening of anti-quorum sensing activity, then you must have Chromobacterium violaceum as the test organism. It's an indicator organism for quorum sensing activity, anti-quorum sensing activity. Okay, so there you have it. That is Chromobacterium violaceum. They really have a very beautiful culture in Luria-Bertani medium because of the violet pigment. And then we also have here Comamonas and delphia. So these two genera are ubiquitous in soil and water. They have been found in hospital equipment and fluids, and they are rarely isolated from clinical specimen. Okay, so they are catalyst and oxidase positive and are able to reduce nitrate to nitrite. Now, I've been mentioning in previous slides the OF medium. So what is an OF medium? Okay, so the term OF refers to oxidative and fermentative. Okay, so usually we have two sets of tube. So we have two sets of tube. So one set has a mineral oil and another set do not have mineral oil. So here it doesn't have mineral oil. Okay, so as you can see, so with mineral oil, no mineral oil. With mineral oil, no mineral oil. Okay, and why do we add mineral oil? Mineral oil on top of the tube, we need to say the tube with mineral oil has no oxygen. And fermentation takes place without oxygen. Okay, so are you getting my point? So fermentation is taking place without oxygen because fermentation is defined as anaerobic respiration. Whereas, obviously, when you say oxidation, this should have oxygen. Okay, so how to interpret? Um, Yellow is a positive result. So as you can see, tube A, both of them are yellow. So tube A is oxidative and fermentative at the same time. Tube B is only oxidative, but it is not fermentative because the one with mineral oil remains green. So it means that it wasn't able to break down the carbohydrates component in it because of the presence of the mineral oil. The presence of mineral oil here means that there's no oxygen in this particular tube. Now, tube letter C, it is not fermentative, it is not oxidative because it remains green. So meaning to say, glucose is not metabolized or they are inert. Okay, so glucose is not metabolized or they are inert. Okay, so that is the OF medium. So I hope OF medium becomes clear already. Okay, so the addition of mineral oil devoids oxygen in a certain tube, and that's the only way for us to assess whether they are fermentative or not. But for oxidative properties, you do not add mineral oil on that particular tube. Okay, so That is the OF medium. So moving on, let's now discuss Flabobacteriaceae. So Flabobacteriaceae is a contaminant of hospital equipment, and they are important cause of nosocomial infection. So the isolate initially appears to be non-fermenters, but they find out that they should be probably be considered as a weak fermenters because the reason for that is reactions are usually delayed. So one of the most distinguishing characteristic of the family is that they are able to produce a lavender green discoloration of the media. So this is the one that are found in nutrient agar. So aside from that, they are DNase positive, oxidase, gelatin hydrolysis, and weakly indole positive. Okay, so that is the general characteristic of the family Flabobacteriaceae. And the members, there are many members. So, so this includes the Balneatrix, Bergiella, Chrysobacterium, Elizabeth kingae, Empedobacter, Myroides, Wixella, Waterciella, Singobacterium, and several unnamed CDC groups. Okay, so Flabobacteriaceae. And initially, they are a large, diverse group of gram-negative, weak to non-fermentative bacilli belonging to a single genus. So they thought we only have Flabobacterium. However, as what I mentioned, members are now separated into a several genera. So all members remain to be indole negative and are not found in human specimen. So the first in our list is Elizabeth kingae, particularly the species Elizabeth kingae meningoseptica. So the cause of most Flabobacteriaceae-related diseases. So Elizabeth kingae is associated with meningitis and septicemia of neonates. And imagine, even a povidone iodine pods could still be contaminated with this organism. So that's why there was a case in US FDA that they even recalled products with this particular iodine because of the contamination with Elizabeth kingae. So they are known to cause pneumonia, endocarditis, bacteremia, and meningitis in adults. Okay, so that is Elizabeth kingae. Okay, so here's how they grow also. So um, in blood agar plate, uh, you would see that they cannot grow on McConkie. Balneatrix alpaca. Okay, so another Flabobacteriaceae family. So these organisms are curved straight rod that are motile by means of a polar flagella. So when you say polar flagella, so there, okay. Colonies are pale yellow and becoming brown as they age. So they cannot grow also on McConkie, and they are also oxidase and indole positive. They are saccharolytic, which means that they can oxidize glucose, mannose, fructose, and other sugars. So the motility and the nitrate reduction should help us to differentiate it from Elizabeth kingae. And then Chrysobacterium endologines, considered to be insignificant in rare numbers, or from one, if and if they are also insignificant if you isolate them from one medium or from one specimen. Okay, so they are linked to nosocomial infections such as bacteremia. So again, when you say bacteremia, it's the presence of bacteria in the blood, or ocular infections or the eye infection. So the source of infection thought to be poor sterilization procedures. Okay. So this is an example uh, of Chrysobacterium endologines as it grows with yellow pigment in nutrient agar. Okay, so Chrysobacterium endologines. Okay. And then Waterciella and Myroides. So Waterciella species are isolated from blood and surgical wounds. Myroides odoratinus let me pronounce it again. Myroides odoratiminus. Odoratiminus. Okay. So they are environmental organisms and have very low virulence. So Myroides is a non-fermenting gram-negative bacilli, and they can cause UTI, endocarditis, ventriculitis, cutaneous infections in immunocompromised patients. So take note of the species, odoratiminus, meaning to say, this would have a very distinct odor as you cultivate these particular organisms. And then Wixella and Sphingobacterium. So Wixella is a species that can be found in genitourinary specimen and can cause cellulitis, tenosynovitis, septicemia, pneumonia, and meningitis. So these particular bacteria may also be associated with dog and cat bite wounds. So pag nakagat pala ng aso, so can actually, your wound can actually get contaminated with Wixella. While Sphingobacterium may cause septicemia with peritonitis. So imagine, peritonitis is actually an infection or inflammation of the body cavity, and perhaps the infection in the body cavity got out of hand, and the bacteria started invading the bloodstream as well, hence the term septicemia. And in the case of meningitis in a premature birth with suspected cellulitis. So, it is isolated from blood and in the urine. Okay, so that is Sphingobacterium. And then, ito naman yung mga kakampinks natin. Okay, so we have the Methylobacterium. So, Methylobacterium is actually isolated from bacteremia, peritonitis, synovitis, and skin ulcers, and they are generally in immunocompromised hosts. So we can actually isolate Methylobacterium from contaminated water. Okay, so this has been implicated in the source of infection. So Methylobacterium is sometimes known as the PPFM. Okay, so talagang kakampinks talaga to, itong Methylobacterium, because PPFM stands for pink pigmented facultative methylotrophs. Okay, so which means that they're able to metabolize a substance with methyl group. So that's why they are called methylotrophs. And as they do, they produce pink pigment. So that's why the nickname for Methylobacterium is PPFM. Okay, so Pseudomonas mucosa. So this one is another catheter-related blood infections and most commonly seen, okay, in this particular kind of scenario. So Pseudomonas mucosa. Okay, so both genera will produce pink to red pigmented colonies like the one that you can see here in the agar. So although Roscomonas would have a more intense pink color. Then we also have Ralstonia and Cupriavidus. Ralstonia piqueti is a contaminant of a sterile fluid. So this can be isolated from urine, nasopharynx, abscesses, wounds, blood, and usually a colonizer or contaminants. So that is Ralstonia piqueti. Cupriavidus focula is another opportunistic pathogens that can cause serious infection. So they are implicated in septicemia, which means in bloodstream, peritonitis, or in body cavities, abscesses, and tenosynovitis. So tenosynovitis is actually when your tendon in the joints are actually infected with bacteria. And one way of confirming whether you have a tenosynovitis would be by means of collection of synovial fluid. Okay, so generally isolated in immunocompromised patients. So collection of synovial fluid, by the way, is known as arthrocentesis. Schwannella, okay? Particularly Schwannella putrescens and Schwannella algae. Okay, so these are infrequent isolates and they are rarely pathogenic, though they are also implicated in abscesses, traumatic ulcers, otitis media, ocular infections, osteomyelitis, peritonitis, and septicemia. So these particular organisms represent colonization rather than infection. So magkaiba yung colonization or infection because colonization simply means that the organisms has chosen a particular site of your body to be its residence. Okay? So Schwannella putrescens is found in stagnant water, natural gas such as petroleum, brine, and spoiled dairy products. So Schwannella algae are more frequently found in clinical specimen as compared to Schwannella putrescens. Excuse me for that. Okay. Singlemonas. There are two potentially clinically significant species of Singlemonas, and these are Singlemonas possimobilis and Para-possimobilis. So they are found in water sources, including swimming pools, hospital equipment, and laboratory supplies. So there are several documented Sphingomyces possimobilis infections. So one is peritonitis associated with CAPD. So again, the chronic ambulatory peritoneal dialysis patients, and then septicemia, meningitis, leg ulcer, empyema, splenic, and brain abscess. So Sphingomyces paraposimobilis have also been isolated from sputum, urine, and vaginal specimen. Okay, so that is Sphingomyces. Sphingomyces do not grow on McConkie agar, but they require more than 48 hours culture on Saborod's blood agar. So as you can see, they can produce this yellow-orange pigment as well. Okay, so that is the Sphingomyces colony. Okay, so there you have it. That's the end of our lecture. So that's the end of our lecture for the non-fermentative gram-negative bacilli.